Swayam Prabha. Digital India. Educated India.
Hi everyone, welcome to the structural biology course again. We are continuing with MD simulation, and from the previous class we are discussing the process to run a sample through MD simulation, which is MD simulation process. So today we will discuss the next part of the MD simulation process. In the previous class we have prepared protein underscore solve underscore ions dot grow. You see that is the setup where you get the protein, you get the solvent, you get the box generated, and you get the ions. Now we will proceed from that, but before going into, there are certain things which should be discussed in detail, and one of them is periodic boundary condition. So first of all, why we need boundary? This is a representation of a protein structure where you see the protein is there, but it is extensively solvated. This is a structure which actually is coming from the like RCSB. So it is a experimental structure, and if you look at the zoomed version, you will see that there are some water molecules which are essential for the interaction with the macromolecule. There are other water molecules which I have circled in red, they are not essential. So when you are going to set up a process, you have to create a cutoff. Otherwise, if you see literally, there is one water molecule which is suppose interacting with a amino acid. Now some water molecule is hydrogen bonding with this, then other water molecule is hydrogen bond with that, then it would be continuing. So your limit would be infinity, but you need a finite system, and that is why we need boundary. What is periodic boundary condition? Periodic boundary conditions are a set of boundary conditions which are often chosen for approximating a large system by using a small part called a unit cell. So here, as I told, you do not want an infinite system to create, because larger the system, more the computational power. So you want to make it smaller, and for making it smaller, you cannot create a real boundary. So you make an imaginary boundary by making some boundary conditions. Periodic boundary conditions are mostly used in performing computer simulation and mathematical models, as we are going to use here. In molecular dynamic simulation, they are usually applied to calculate properties of bulk liquid, crystal, or mixtures. A common application uses periodic boundary condition to simulate solvated macromolecule in a bath of explicit solvent. We have already discussed about explicit solvent. There are explicit solvent and implicit solvent, and the process we are going through, the MD simulation process, we are continuing here. If you remember, in the last class I talked that we have selected TIP3P water molecule, which is part of the explicit solvent model. So there are very interesting problems coming in accord of choosing periodic boundary condition. Let's say you choose periodic boundary condition and you choose a single unit cell. Here is the run where a single unit cell of water molecule is chosen and a protein kept in the center of the box and the simulation run developed. If you look at after 127 picosecond of the run, the protein reached kind of at the corner. At 304 picosecond, it is already out of the solvated system, it is already in vacuum. In 500 picosecond, it is far away. In 700, it is even maintaining more distance. So, this is not possible. So, what you have to do in addition to applying the boundary condition, you have to develop a central simulation box and then you have to replicate that around. You have to make layers. Okay. Now, you have to remember, as I told, they could be going from the central to the other one, as you could have seen here. The central simulation box is extended to infinite dimensional system by considering its periodic images. Now, it is not the problem. Here, if you look at the blue particle, this one has neighbors within a radius, if you see, which is represented by light orange circle, which are periodic image of particles from the boxes, these boxes, which are light green circle cuts. So, in that way we set and we run the simulation without having problem. Now, I come to another very important component of simulation run, which is called phase space. First, to define, in dynamical system theory, a phase space is a space in which all possible states of a system are represented, with each possible state corresponding to one unique point in the phase space. Let me explain a little bit. Before that, for a model system, the phase space usually consists of all possible values of position and momentum variables. So, for the model system, we are looking at the position and momentum. So, what should be the starting one? A coordinate, which we have already discussed. Let us look at a model. A given atom is represented by its time dependent position and momentum vectors. This is the IMO atom. At a given instant, consider a plot of the position and momenta for all the n atom in the system. So, we start with the I atom. Now, we are considering all the n atom. So, there would be degrees of freedom in terms of position and momenta. So, there would be 6 n dimensional plotting domain, and that 6 n dimensional plotting domain define the phase space. Let me go into more details little bit to so that you could understand, but before that I have to tell you the concept of ensembles. Phase space and ensembles are very correlated. Before going into, I will take you to a previous slide which I have already discussed, the 3 systems: in vivo, where I told there are mixture of proteins; in vitro, where you have one protein but in many numbers; in silico, where you have proteins depending on how many you want, one, and second is you have the coordinate information. So, we have taken out the in vivo system from this discussion because this is complex. 1, 2, I have already discussed about the fact that when we are talking about the dynamic system, the MD simulation modeling, our goal is to mimic a in vitro system and not an in vivo system. So, now if we look at a in vitro system, the many molecules they have a trend of behavior, and here we are taking one. So, we are actually making a journey of ensemble. I am coming to discuss it in the next slide. In addition, here mostly we do not have coordinate. We cannot control coordinate in control or operate coordinate in vitro experiments. Whereas, in dynamic modeling or any computational study, we could control the change of the coordinate. So, as I told, now I come to the concept of the correlation between the phase space and ensembles. So, if you think about a system, let us say about us, one person, entirely we represent a macroscopic system and our cells are representing microscopic system. Even smaller the protein, if it could be in a crystal, the crystal is a macroscopic system, individual protein is a microscopic system. So, a macroscopic system could be divided into many microscopic system, and these are called statistical ensemble. Now, the property of the macroscopic system is average property in all the microscopic system, which is called ensemble average. As I already told, we could have controlled the coordinate of our sample while doing dynamic experiment. If you look at phase space usually consists of all possible values of position and momentum variables. In the coordinate, you have the position, and when a force is applied, it gets momentum. So, you have position and you have position change information. So, we could have go into the phase space. In that way, we have connect the ensemble with the phase space. So, what is an ensemble? An ensemble is a collection of all possible systems which have different microscopic states, but have an identical macroscopic or thermodynamic state. I repeat, collection of all possible system which have different microscopic states, but identical macroscopic or thermodynamic state. There are different type of ensembles which exist with different characteristics. Microcanonical ensemble is NVE. The thermodynamic state is characterized by fixed number of atom which is designated by N, a fixed volume V, and a fixed energy E. Canonical ensemble NVT. This is a collection of all systems whose thermodynamic state is characterized by a fixed number of atoms N, a fixed volume V, and a fixed temperature T. Isobaric, isothermal, isobaric and isothermal ensemble. Isobaric is pressure, thermal is T. So, NPT. This ensemble is characterized by a fixed number of atoms, a fixed pressure and fixed temperature. The last one, grand canonical ensemble, mu Vt. The thermodynamic state is characterized by fixed volume, fixed temperature Vt, and a fixed chemical potential which is defined by mu. So, we have discussed periodic boundary condition, its role, we have discussed phase space, and we have discussed ensemble. Now, we will discuss about another thing which is again very critical, probably the most critical thing in biology, which is energy minimization. Before going into biology, in general, energy minimization in the field of computational simulation It also called geometry optimization is a process to find an arrangement in space of a collection of atoms where according to some computational model the net interatomic force on each atom is acceptably close to 0 and the position on the potential energy surface is a stationary point. So, I do not go into the second part now, I am going to explain it, but first part is you have a atomic system, it have bond length, bond angle on all these things, it would be optimized in a way, the configuration, conformation, geometry, that the net interatomic force on each atom is acceptably close to 0. That is the major condition of energy minimization. The collection of atoms might be a single molecule. Nowadays single molecule is becoming very popular for study. An ion, a condensed phase, a biological macromolecule, a transition state, or even a collection of any of these. The computational model of chemical bonding might, for example, coming from the calculation of quantum mechanics, whereas computational model of protein movement coming from Newtonian physics. When we come to protein, as I told, minimization have a huge role in studying protein. We will go back to Anfinsen's theory. If you remember, when I was discussing protein, I have talked about this, how Christian Anfinsen have changed how the world is looking towards the process of protein folding by a simple experiment. Before Christian Anfinsen, people used to think that the folding of protein is a biological phenomena, which means the interaction between the atoms, the charge, the size, all these, if you think molecularly, you will clearly understand there is physics, there is chemistry. But before you have those molecular concept, people used to think that this folding is a biological phenomena. With simple experiment, he had proved that it is a physico-chemical affair, and that information changed the way people have studied protein. Before, people wanted to purify a protein only from the inside, only from the biological system. If you look at, I talked about this before, if you looked at, before people have, most in the primitive time when people have started working on protein, mostly they work on protein coming from blood, myoglobin, hemoglobin, because they have collected the blood and work from them. But Christian Anfinsen's theory, his experiment, which definitely gave him Nobel Prize, have changed the idea the world have. So, how we could do energy minimization? One thing is to understand the potential function, then evaluate the energy of protein conformation. Before, people used to know that every protein have one conformation, but now we know that protein for functioning change their conformation. So, we have to evaluate, we have to select the native structure and go from them. Conformational search algorithm to produce new conformation, search the potential energy surface and locate the global minima. So, the potential energy surface, which I did not talk about, again coming. So, let us first discuss about potential energy surface. So, potential energy surface is a map. Okay? You will see here in the x coordinate there is a variation in parameter, and in the y coordinate there is potential energy. So, a potential energy surface is a plot of the mathematical relationship between the molecular structure and its energy. So, in one side there is energy, in one side change, what is defined in the variation in the parameters. The surface might define the energy as a function of one or more coordinates. If there is only one coordinate, the surface is called a potential energy curve. So, what you are looking at here, x and y axis is given. So, it is a 2D curve, but it could be multidimensional also. A potential energy surface is a conceptual tool for aiding the analysis of molecular geometry and chemical reaction dynamics. Once the necessary points are evaluated on a potential energy surface, the points can be classified according to the first and second derivatives of the energy with respect to the position, which respectively are the gradient and the curvature. So, you could have get the trend, how it is happening, where it is happening. Let us discuss here. So, at the first, the position is a starting point conformation. Now, if you apply minimization, if you do the lowering of energy. So, when we are talking about, one thing I should go back and talk again is the words I did not utter here. Protein's native structure correspond to the state with the lowest free energy of the protein solvent system. That is a hypothesis which is giving us a proper drive. So, we know that if we have a curve like that, definitely the lowest point would be considered as global minima. So, coming back to that, so this is your starting point, you apply energy minimization, you come to the local energy minimum. There are other points, one I already talked about, global energy minima. Another one is transition point. Also, this is energy barrier. Transition point is important, especially for people who are studying chemical and enzymatic reactions. Identification of transition state is a very critical thing in the field of drug designing, in the field of understanding a reaction and its further proceedings. So, if you see, I had shown you a very simple potential energy surface. It could be much complicated, and in case of protein, it is a funnel like, which we have already discussed. When you are talking about potential energy surface, to measure it, we perform the derivative calculation. The first derivative, which is dE dx, if that is 0, then we know that we have reached a static or stable point, which you could also talk about as maxima and minima. But then when we go for double differentiation, d2e dx2, if d2e dx2 is greater than 0, then it is maxima, whereas when you are getting d2e dx2 less than 0, then it is minima. But among minimas, if you see, these are local minimas, whereas these represent global minima. As I told, the stable states, the minimum of the potential energy surface with respect to every coordinates. The transition state or the saddle point is minimum in every normal direction, but one for which it is maximum. Stationary points thus correspond to important states of the system. If you see, for minimum d2f dx2 greater than 0, d2f dy2 greater than 0. For maximum d2f dx2 less than 0, d2f dy2 less than 0. For saddle point, d2f dx2 greater than 0, but d2f dy2 is less than 0. So, by this double derivative we could identify the maxima, minima and the saddle point, which is the transition point. As I told, in practice there are many dimensions of the surface, there are many local minimas, and that is why we have to perform energy minimization. What energy minimization is going to describe? Either a molecule or ensemble of molecule having constant atom composition, a system where a chemical reaction occurs, relative energies for conformers, and an example I am not giving you now because we have already discussed about that. The conformation of N-butane as the global minimum is the anti-conformer, local minima are the Gauch conformer, and the saddle point are the eclipse conformation. What they are talking about is, I hope you guys remember. So, if you have a molecule like this, because of these two sterically bulky group being at the farthest position, they are showing the most stable state, which would be defined as the global minima. Whereas, when they are close, this would be represented as the global maximum. Energy minimization is a numerical procedure for find a minimum of the potential energy surface starting from higher energy initial structure. If you look at this picture, during energy minimization, the geometry would be altering in a stepwise fashion. Energy, so you see here the first one is at high energy and you are going down in energy with the alteration in the geometry. So, during energy minimization, the geometry would be altering in a stepwise fashion. Energy of the molecule will be reduced from 1 to 2 to 3 to 4 in that fashion. So, for the determination of energy minimization, most of the energy minimization method proceed by determining the energy and the slope of function at point 1. If the slope is positive, it indicates the coordinate is too large. If the slope is negative, the coordinate is too small. After that, you could optimize further with numerical minimization technique. The numerical minimization technique then adjusts the coordinate. If the slope is positive, it indicates the value of the coordinate is reduced 0.2, it comes to 0.2. If the slope is 0, a minimum has been reached. If the slope is still positive, coordinate reduced further 0.3 until a minimum is obtained. So, this is the search towards getting minimum. All the energy minimization method used to find a minimum on the potential energy surface of a molecule use an iterative formula to work in a stepwise fashion. If you remember, this is one thing I have talked about continuously. Iteration is what a computer doing for you. These are all based on formula of the type x new equal to x old plus the correction. In all these methods, a numerical test is applied to the new geometry to decide if a minimum is reached. There are two types of energy minimizations: first order minimization and second derivative method, second order minimization. First order, two popular methods: steepest descent and conjugate gradient. Second order, it is Newton-Raphson. Steepest descent, the second derivative here is assumed to be constant. The equation to update the geometry becomes x new equal to x old minus y e prime, where y is a constant. In the steepest descent, if you see the picture, it starts from the derivative vector of the initial point, define the line search direction. So, it could directly go there, but it do not do that. The derivative vector does not point directly toward the minimum, the minimal point. The negative gradient of the potential energy, the force, points into the direction P0 here to B, and then P1, it reach P1 by going in the direction of B, it reach P1 and then go to C, which is perpendicular. So, the negative gradient of the potential energy points into the direction P0 to B, then P1 to C of the steepest descent of the energy hypersurface, and is always oriented perpendicular to the energy isosurface. So, it comes in one direction, then go perpendicular, then comes in another direction, go perpendicular. In that way, it reach the minima. The method is named steepest descent because the direction in which the geometry is first minimized is opposite to the direction in which the gradient is largest or the steepest at the initial point. In this method, gradient at each point calculated, no second derivative calculation required. The method is much faster per step, more steps required to find the minimum. The main problem with the steepest descent method is determining the appropriate step size for atom movement during the derivative estimation steps and the atom movement steps. Coming to the next, the conjugate gradient. This is also a first order minimization technique. It uses for both current gradient and the previous search direction to drive the minimization. The number of computing cycles required for a conjugated gradient calculation is approximately proportional to the number of atoms. So, with increasing number it increases, and the time per cycle is proportional to n square. It improves the step efficiency. The method takes the next search direction to be a linear combination of the current gradient and the previous ones, require fewer energy evaluations and gradient calculation. Convergence characterization are better than with steepest descent. Coming to Newton-Raphson minimization method, this is the most computationally expensive per step of all the methods utilized to perform EM because of the second derivative. It is based on Taylor series expansion of the potential energy surface at the current geometry. You have already see how we use Taylor series in different cases. The equation for updating the geometry is x new equal to x old minus e prime x old by e2 prime, the double derivative x old, is a powerful and convergent minimization procedure. The advantage of Newton-Raphson procedure that the minimization could converge in one or two steps. The major drawback is that this method requires the calculation of the second derivative, so computationally expensive. The minimization can then become unstable when a structure is far from the minimum or the energy surface is enharmonic. So, advantage: only one iteration for quadratic functions, efficient relative to first order methods, better energy estimate. Disadvantage, n square storage requirement compared to n for conjugate gradient because of the second derivative, involves calculating Hessian, which is 10 times more gradient calculation. It used in transition structure searches, which we have shown the saddle point. Saddle point, as I told, is very important for identifying the transition state of a reaction or the transition state or intermediate of a biochemical reaction. We are coming back to the process estimation. So, we would look at the steps in MD simulation. First, we have talked earlier, this is the initial coordinate which is prepared for simulation. But as I told earlier, from this initial coordinate you could not directly go to simulation. What you have to do? You have to do a lot of processes like adding hydrogen, coming to structure 2, then you have to develop a new box, then you have to solve it like add water, and finally you have to add ions. So, this is where we have finished last lecture. Now, when we come here, it could possibly, the structure we have, it is process now, but it could possibly have bad contacts causing high energy and forces. So, there is a requirement of energy minimization. So, the next step in MD simulation would be energy minimization. So, coming to the flow again, we have the initial coordinate, we have processed them, we come to the structure where we have the protein, it is now having a box, periodic condition is applied, solvated with TIP3P, ions are there to bufferize. So, everything is there, but now it could possibly have bad contacts. So, we have to go for minimization. So, we have to perform energy minimization, it would help finding the nearby local minimum. So, if you perform minimization, you reach here. What next? From this local minima to another local minima, you need equilibration. We talk about ensembles. Now, these ensembles would be used to equilibrate the sample to escape the local energy barrier. Then from there we will come near to the global minima, right, and this would be done by the classic simulation samples. We will get the thermally accessible states. So, these are mostly like globally minimized, we could see the movements and all, but one thing you have to understand here, I say classic simulation. Why? I will talk in the last class of this module. So, with protein underscore solve underscore ions dot grow, we have taken these three. If you remember, it is a similar process, the minim.mdp, the .grow and the .top file, they are converted to .tpr using the command grompp. Here, steepest descent minimization method used, we have already discussed what is a steepest descent minimization method. Now, we are, as I told, actually going to the MD run, the minimization run, and after the MD run we get em.grow, the GrowMax competent file. But in addition to the em.grow, we will also get em.edr, em.log and em.trr, the new files. Let us take a look. Files with the TRR extension contain the trajectory of a simulation. So, TRR are files which are giving you the trajectory. Remember, I told when you have a like any sample, you put it on the force field and you apply force, it would go up, up, up and then come down. So create a trajectory. In this file, all the coordinates, velocities, forces and energies are stored. What is .edr? The EDR file extension stands for portable energy file. So it would record the energy change. The energies are stored using the XDR protocol. em.log file contains detailed information on several energy terms throughout the simulation and then a performance analysis at the end, which we will see in the analysis part. .grow contains the coordinates of the final configuration of the system. So now we minimize it. Let us take a look what happened. So here what we have done, we have aligned the energy minimized structure with the initial structure we have started to look at what are the changes, and if you take a zoomed look you will see that there are changes here, but especially when you pick up structures from the experimental database you do not see much changes, but you see changes here, whereas in other part you do not see that many changes, but there are little changes also in. So changes are there but not much. This is clearly showing you how from the high energy the molecule is minimized. So our energy minimization is successful. In that way we could check the energy minimization. What next? The equilibration. So we get the minimized file em.grow. We again do the same thing by applying GROM-PP. We come to nvt.tpr. So now what we are doing? We are doing nvt. If you remember, this is an ensemble. What we are getting here after the md run, we get nvt.grow and is the main file. Additional files are nvt.cpt, nvt.edr, nvt.log, nvt.trr. Most of the files I have already talked about. The TPR file we already told also about that. It is stands for portable binary run input file. It contains the starting structure of the simulation, the molecular topology and all the simulation parameters, and because it is in binary format it cannot be read with normal editors. You have performed 100 picosecond run for NVT equilibration as I told, and this is what you get after NVT, that how equilibrated this is. This is a plot between temperature and time. Now you come here with nvt.grow, again you do the same thing with npt.mdp because you are going to do equilibration with npt condition. So npt.mdp, nvt.grow and topol.top, 3 files again merge with GROM-PP to give npt.tpr, and then you again do the md run, you get npt.grow. With that npt.cpt, npt.edr, npt.log and npt.trr. So I talk about all those files except this npt or nvt.cpt, I will talk about that. This is again a 100 picosecond run performed for npt equilibration. So this is the curve you get when you put pressure versus time. To compare, I put the other file, so you could see that this is NPT, this is NVT. Okay. The CPT file, we will talk about this in the last, but it is a checkpoint file. What is a checkpoint file? A checkpoint file helps you to run again if your simulation is stopped because of any other reasons. Now we are on the final stage. We have this npt.grow file, again we have npt.grow with updated topol.top. So npt.grow, topol.top and md.mdp. These three with CROM-PP make md_0_1.tpr, and when we do mdr, here we get .gro file, .trr file, .cpt file, .edr file, .log file. In addition, we get .xtc and previous.cpt. Previous.cpt, this file generated to take a backup, because this is 100 nanosecond run. If you say 100 nanosecond final MD simulation run performed for each system. So, if anything happened, if any problem, before it was like 1 hour or something, now it is like 6 days, 10 days. After the end of 10 days, if you have to rerun it, it is really a loss of time. So, they make a backup. So, they make a checkpoint file, they make a previous checkpoint file one. Then think about, if you are saving, remember I talked about every 1 or 2 femtosecond, in 100 nanosecond, how many files are created? That would make the .trr, the trajectory file, hugely big, huge. So, while .trr is a very big file now, unzipped, you get .xtc, which is having same information but zipped, so much smaller in size. So, we have run the MD now. What is the result now? You will see that it is dynamic. It have run, so you will see different conformers of the sample. That is what is the end product of a MD simulation run. Now, from there we will go to analyze the system, as I talked about. From this we will get a lot of information according to how we have set up the experiment. We have talked about process. Always remember, process details have no role in your result. It is how you set up the experiment, you get the result out of that. I will talk about that in details in the next class. Thank you very much for listening. This is relatively longer period of time I have taken, because I wanted to finish the MD simulation process. But you guys are amazing. Thank you very much for listening. Thank you.